Thank you so much for coming through. It gives me great pleasure to introduce to you our speaker for today, Professor Nicolas RABET from Sorbonne University in France. Nicolas has been with us in South Africa for the last 14 days, 12 days, and we've been throughout the country collecting streams, and as part of the South Africa-France collaboration, Professor RABET is a full professor at the University of Sorbonne in Paris. He did his studies in Evo developmental ecology in the laboratory in Paris Cité, and then did a postdoc in molecular biology center at CNRS, which is an important institution in France. And now he works at a laboratory in Sorbonne University, where he does work on various crustaceans. We are so delighted to have Professor RABET, and I would like to hand over to him to give us his lecture. Over to you, sir.
Thank you. Thank you very much, Musa. Just, I will make a lecture about hydrobiological collaboration between France and South Africa. New approaches for study freshwater. Before to start, I have to say a very huge thanks to Musa and Sam, because we made something, a project, and it was a very lovely day in South Africa, and I'm very happy. Then it's a real pleasure to introduce my work, and I will try to explain a little what we are doing in South Africa and what we can expect for future in relation with this collaboration. Okay. Firstly, I have to thank also the program who paid this work. The code that we obtain is PROTEA. It's a combination of French campus rules and the National Research Foundation in South Africa. Then we obtained money for 2 years, and the first year was for us to come in South Africa, and next year some South Africans should be in France. Firstly, this project conduct to make meeting about firstly distancial meeting and remote meeting, but also we made several meetings during the trip in South Africa. Then we discussed about different aspects of our research, and we combined what is possible to do in this collaboration. It's very nice. The South African partners organized a field expedition. You have the map, then you know the size of South Africa. For the French people, I have to say that we make a lot of cars, and it's sometimes horrible. But the country is absolutely amazing, and we are very, very happy. Valentin and me. Valentin is not here because he is in the Réunion Island, but we have he cannot speak. But here the presentation, and we're very happy to be there, and this organization is very nice. Then we travel in different places of South Africa. We make visit of collection in South Africa, the Kruger Museum, but also Albany Museum. We saw many interesting things, and relatively, we work in Museum of Paris. Then it's very interesting to see the potential collaboration at the level of collections. Also, we make different observations. We make sorting, learning students, and we make also some determination. The work was relatively huge. In fact, we worked not a lot, but it was absolutely great. We saw two animals, two types of shrimps. Firstly, before to start, I have to introduce the shrimps. We worked in two types of shrimps: the carrot shrimps, who are the true Malacostraca, and large macropod shrimps, who live more in temporary water. Then we have two types of organisms, two types of habitats. Then we worked on temporary ponds of large census, and also rivers. And for French, I had some different things. For you, it will be normal, but this is a crocodile. Then you have to know that in South Africa, we cannot go in the river like that. We have to take a lot of precaution. This is a crocodile river at the entrance of Kruger Park. We have a lot of good times. We make something, I think, interesting at the level of human relation. It will be, for us, it's very important to start a collaboration with this type of relation, because the work is hard and we need to have good relation. We make a lot of things interesting. You see also the giraffe who make a smile to Valentin. We were in the other card, and but this is for this year. Next year, some South African guys should go in Paris. Then I make a small map to you to learn now. This is the Sorbonne Université campus Jussieu. Then this is sort of a square, very French organization. And you have a museum very near that you have just to cross the road. And also, I think that you will try to go with a TCA summer meeting that we organize in France. And Valentin and I are in the organizer committees, and it will be interesting. I had Mona Lisa, because we have a lot of discussion about Mona Lisa and the Eiffel Tower, where you will, of course, be invited in a BOREA lab. What for the next year, you have to visit the collection in Paris, Zootech. You will see. Don't afraid, the collection of crustaceans is not huge like that. It's only the mammals and things that take a lot of place. And also, we will discuss about that, but I previous to do some stuff on microscope and scanning electronic microscope, who are represented here, to see eggs and animals in detail. What is the new approaches for Swedish freshwater shrimp that we try to elaborate together? It's firstly, we make an interrogative taxonomy, who a combination of morphology and genetic, geometric, morphometric, ecology, and distribution of species to give us good as possible stature for the species. And we are absolutely sure that this integrative taxonomy should do with a collection like Albany Museum collection, Natural History Museum and in London, and the French Museum, because I think that the majority of animals from Africa are in this two institutions. Then this is very important to do this work in relation with capacity to this different museum to learn and to accept to register more animals from this type of field. As you see, there are different aspects about taxonomy, and we try to do as possible modern taxonomy in order to improve knowledge, especially in South Africa. But the problem is not limited at the border of South Africa. But at least we have to start somewhere. In Africa, I think South Africa is the best place to start. Valentin learned electric fishing for fishing shrimp in river. That you have some picture of that, and some shrimp are very huge and very nice. It's a microbachium, and I hope, yeah. And you have the nets. Then they make an electric things in the water, and the animals go with the current in the net. Then it's a very powerful technique. That's one thing that we do together. Now I'm more a specialist of large macropods, then I will speak about this subject. Is more my own subject. You see who are large macropods, what are You have a tadpole shrimp. This one is particularly huge. It came from Africa, but not at all in South Africa. It comes from Morocco. It's the largest species that I never seen for tadpole and probably bronchiopods. More than 15 centimeters. It's a huge animal. Also, you have clam shrimp, fairy shrimp. Fairy shrimp is a very elegant animal that people like in general. And clam shrimp are the forgotten animals because they are small at the bottom and more or less dark. But in clam shrimp, there are many things: Spinichordata, Levichordata, Cyclesterida, and this group is not a good group at the level of a taxonomy, but we continue to use this word because it means something on the field. How does this type of animals can survive in temporary ponds? You see dry Olinnadia that I saw in Martinique in French West Indies, and you have a tadpole shrimp from France dead at the bottom of the pond. How they can survive, and they use a strategy the resting eggs, who are accumulate in the bottom of the ponds and in the sediment, and create a sort of egg bank, who are regularly used, like for plants with seeds. All the seeds cannot germinate at the same moment. It's the same for resting eggs. I made some work on paleontology, and I have just a remark relatively to that. This is the first Notostraca tadpole shrimp that we saw in the world. It came from the late Devonian, and these animals are with other animals that you can see. They are already clam shrimp, but also fairy shrimp. And we found in the fossils the eggs, meaning that this strategy of production of resting egg is very old. Then, if you placed back in time all the large macropods that we know, at the early Devonian, there are very few animals, and they are very different morphologically. At the end, at the late Devonian, they look like the modern one. And we have few registers in the fossil to understand the evolution, but some people say that there are living fossils, but this is not acceptable because if you are fossil, you are not living. But just the idea is that the animals appear at the Devonian during a short period of time, and after the morphology didn't change a lot, and it's a morphologically stasis. But it doesn't mean that the animals don't change; they change a lot. But the general morphology are more or less the same. And something very interesting: this strategy give a capacity to live in temporary water and continental area since the late Devonian. The late Devonian, it's the period of the first season tree, things like that. It's a very important first step of the true occupation of the continental area. Then the large macropods are in the same in the first wave, and where they live, they can live in polar climate, in desert, in temperate climate, and tropical. In polar, you can imagine that it's not the dry, it's more the freeze to make that the water are not disponible for the animals, available for animals. In South Africa, it's not the polar climate, there's more with so many savanna ponds, and just also for French, but I had different observation. We are not alone in the savanna ponds. One time we are in front with a hyena. Another time it's a giraffe. Giraffe is less problematic, but you saw that we are obliged to have a guard with a gun because it's not so easy. It's not like crocodile, but it should be difficult. We can be a little ridiculous in the ponds, but I accept the idea. Then we sample a lot, and the water is very clear, like you can see, not completely muddy. You are be careful to don't go to the water, if not you will look like the pool. And also we studied rock pool, but I make a the comparison between French and South African. And where are the French? Here, I put at the left, and South African in the south, in the opposite. And if you look at, they are very similar ponds, and the animals are at the middle. At the left, you have tiny mastics, and on the right, you have Branchipodopsis from South Africa. It's in order that you see that the similarity is very important. The big difference is that in France, we have only one species of ferrucine, but two species. That one species of tiny mastics, and in South Africa, you have different species of Branchipodopsis. Also, we discussed about that. That's why I put the picture. I compare the wetlands in dry environments, and you have a South African one and French one. They are in sandstone the same type of rock. And we found something very similar. Then ecologically, I think we can make a comparison. And it's not so stupid because in Drakensberg, the climate is not so hot, then it's not so different. In expected environments for flower branchiopod, I had to show you different pictures to help all the people from France and from South Africa to understand where we have to look at. Firstly, salinity variable ponds under hypersaline environment is a good one, a good place for large branchiopod, especially ferrucine. We have also some very unexpected ponds in trail or the dirty road, very small. And in Martinique, you have Elinadia who live in this type of ponds. The deep is 10 centimeters, no more than that. And in continental France, you can have different species who live in this type of ponds. This environment is near Paris, it's Chirocephalus, 3 centimeters of length. Also, we can have a very strange environment. Medieval grave could be inhabited by triops. Then it's the people make a hole in the rock, and after the body disappears, and the system doesn't like that, just a hole. But the water can fill this hole, and the animals can move. This is very particular. Also, something that you don't have for the moment. South Africa, we have some tadpoles who live in river, temporary river. Then it's very interesting. It's not in France, it's not limited to the pond. It could be also a small river. Generally, you have an idea of temporary ponds, but not a real idea of the diversity of habitats. This pond is in New Caledonia. It's called the Trou du Tahitien hole. I already go with Valentin and the French TV to go there. The hole, the water can be between 50 and 60 meters of water. Then you cannot swim at the bottom when the pond is full. Very, very original. Also, but not in France, but in South Africa, you have we didn't go in the desert. In the desert place, you have very huge ponds. Very huge. Sometimes you can drive on the ponds during several kilometers before to realize that it's a pond. And in this type of things, you have like a sort of lake, temporary lake, which is also a very interesting environment. What we can study when we can do this in this type of environment, 3 new things that I propose is studying sediment containing eggs, image for satellites, and using drone to improve knowledge. Firstly, of course, net fishing is the first technique, and the traditional methods are always good, meaning that you can, of course, if you have water, you can put your net in the water and obtain the animals. Then this is the end of that stuff. And you can see fairy shrimp, 2 species of fairy shrimp. But also, we can obtain eggs in the sediments. Then you take the sediments in the bottom of the pond, and we can analyze the sediment to obtain the resting eggs. The resting eggs have a size and a shape particular than you have here triops and Streptocephalus. It's not the same shape and not the same size. Here, it's a French point of view of the diversity of egg resting eggs to show at the same scale the variability of shape in France, meaning that, of course, the diversity in France is less than in South Africa. And I propose you to have some idea of shape and the animals and the eggs, but it's from example from France because we just found the animals and we have to do that. Branchiepulse fairy have a head not really spherical. It's with big depression like craterus, and the shape is very easy to recognize in the sediments. You have from New Caledonia pyramidal heads, very typical. Then you have in South Africa some species with pyramidal heads that we didn't find. And this is very interesting because if you saw this type of things, you can be sure that it's Streptocephalus. You have here an example of Chirocephalus, the most frequent species in France, and as the majority of animals of ferrution, the eggs are spherical. And here in this case, they are palisadic structure ornamented the eggs. We have the species that I show you from rockpool, which is a specialist of rockpool, and the eggs are lenticular. This is the only case in the evolution of, I think, of eggs that they are lenticular. If you look at the picture, you can see the shape like the lenticular, some seeds that you can eat. And these animals, typical of fresh water rockpool, are present only in Europe and North Africa. Now we start to speak about Elimnadia. Elimnadia is a very diverse genus with a lot of species living in all the tropic and or sometimes in temperate place also. And in New Caledonia, you have the shape of the animals, and a nice picture of my friend Jean-François K, but also the shape of the eggs. Here is Elimnadia morrisana from Reunion Island. Why I show you is because I think that the species live also in South Africa. And you can compare, you will see it's clearly different, and we can identify the two species like that. And I show you now different species of the same genus. And you have the number is just because I put the picture like that, but they came from different places of the world. And as you see, the morphology of the eggs is very variable. Then we can identify the species, but we can identify the eggs in the soil, in the sediment. Then, which is very interesting because Elimnadia live only 2 weeks. Then it's very hard to find them in the ponds. Frequently, we found only the eggs. It's why we studied that also. What you can do with sediments, you can make breeding, which is very complex and hard. But we can also extract the eggs from the sediment, and we can make a morphological analysis. And we are working about that in the lab now. We try to obtain the DNA without extraction in order to make PCR. Then meaning that with sediment, we can have an idea of the genetic structure of a population, and we can have an idea of a number of species in a pond. It's less expensive because we don't have to make extraction, and we win a lot of time. Then the PCR can be done in less than 1 day from the sediment, which is very rapid. Two other things that we want to improve, not only in South Africa, we are working on that in France too, that we try the idea is to try to do that also in South Africa, is to use Google Earth to find temporary ponds. Nevertheless, we have a problem with Google Earth because the period of satellites is variable. Then we can have a 5 picture of January of a place and no picture during 2 years. Then it's impossible to survey the pond like that. However, we have different picture of the same place. Then I make an example that Musa know very well. I think you recognize. And you have a picture with all the ponds are full of water. Another one, all the pools are dry. And if you look at the picture above, you will see that one pool have water and not the other, meaning that this these ponds are not at the same level of temporality than the other. Okay, it's good, but we can use also drone to investigate the ponds. And I show an example that we did with Musa. Not me, because I'm not more a specialist of climbing in Drakensberg, but we use the drone to go in a place before to go by feet in order to win time and to see if the water are present in the ponds. And you have a picture of the ponds before to go, and Musa can go because he saw that there are water. This is the first application. Two other applications possibly to the drone if we can fly. This is also the problem because you cannot fly everywhere. But you can go to another mountain to see how is the place before to lose 4 hours of walk and climbing, to see 5 minutes later that the water is not present. This is very interesting. We didn't perform that, but I think in future it's a good idea to, and I found 2 ponds, not so far, 2 kilometers, and the drone can do that at 50 kilometers per hour. That means that you will have a result in 5 minutes and not 4 hours of walk in the mountain. Other things, it's not in Drakensberg, but it's a rocky place at 2,000 meters of altitude. This place is relatively dangerous, and we can have a prospection very easy with a drone to see where are the rock pools. And I put 2 red arrows to see the more huge one. And if you look at very well, you will see Daska, the white girl in the rocky place, meaning that the ponds are relatively important in size. Okay, and also, what we can do with a drone, if we combine between drone and Google Earth, you can have, I compare the picture that we obtain at 120 meters and the best image of Google Earth. And you have detail, very interesting. We can easily recognize the ponds, but we can have detail relatively to the disposition of the vegetation and different type of things. And we are at 120, we can make the same photo at 40 meters, for example. But the connection with Google Earth is perfectly acceptable and useful for paper files. Then I don't know how many time I spent, but it's my last slide. And again, I have really to thank my friend. I say my friend, I hope it's for the both sides, but Musa and Sam to help us in your investigation and the idea that we can work together to have a better knowledge about South African wetlands and temporary ponds. I think it's something very important relatively to the climate change that we are that we will live. Then it's something that we have to manage and to use the museum to improve the knowledge, the collections. And I'm very, very happy to be there. And thank you very much. And if you have any question, I will try to respond to those.